In this video, we're going to be talking about enzymes, which are proteins that act as biological catalysts. So I know that this title is a little bit long, but it will make sense as we go through the slides. And this picture is just a random picture of a protein because, as I already mentioned, enzymes are made of proteins. And so this is the tertiary structure of a protein. You're going to see that the shape and structure of enzymes is extremely important in their function. And so that is why I'm showing the tertiary structure, because that really is the most important part of an enzyme. All right, so as I mentioned, enzymes are biological catalysts. So what does a catalyst do? A catalyst lowers the activation energy of some kind of chemical reaction. So in biology, there's lots of chemical reactions happening in living things, to break down food or to harvest sunlight for energy. There's many different things, types of chemical reactions that are happening. And a catalyst is something that lowers the activation energy. So the activation energy is the push that is needed to start a chemical reaction. So I like to think of it as when you start your car, you have to turn that key or push the button, or simply when you're starting a fire. So when you start a fire, even though that is an exothermic reaction, meaning that it is releasing energy, you still have to light that match, or, if you're old school, you're using like a rock and some twigs to start the fire. And then once you start the fire, then the reaction goes. And so the activation energy is this little push that you need to get the chemical reaction started. And when we use a catalyst, this lowers that activation energy. So here's the activation energy of a reaction. If you have a catalyst, that energy is lowered, so it's easier to start that reaction. So again, going back to the analogy with a fire, if we were to start a fire the old school way using sticks and a rock, that would obviously take a really long time. You're putting in a lot of effort. But if you use a match or a lighter, that's obviously much easier, and you're going to be able to get that fire started with less energy. And so in that kind of situation, the match and or the lighter would be your catalyst. Now, what's important about catalysts is that they don't affect the beginning and end of a reaction. So the reactants and the products are unaffected. The reaction will go the same way. So for example, again, if I start a fire using twigs and a rock, I still get a fire at the end. If I start a fire using a match, I still get a fire at the end, right? So the end result and the beginning are unaffected. And again, the whole point of a catalyst is to allow reactions to happen more readily or more easily. And of course, in biology, in a living thing, that is really convenient. And so enzymes are proteins that are found in living things that act as catalysts. Their whole job is to lower the activation energy and make sure that reactions are happening more easily. And so this is how an enzyme works. An enzyme will bind to a substrate, and that substrate reacts. And so enzymes react to the substrates. The substrates bind to the enzyme at this portion of the enzyme called the active site. So here's our enzyme. Here's its active site. And the substrate, which by the way can be any molecule, will bind to it, and then the chemical reaction begins. Notice that the substrates bind to the enzyme at the active site, and that fit is very it's like a perfect fit. So it's like a lock and key. And this again goes back to the idea of, the shape and structure of an enzyme is extremely important. If there is something wrong with the shape of an enzyme, it might not work, because if this shape is not correct, then the substrates aren't able to bind. So it's a very kind of specific situation that's happening. Now, the reaction happens, however it happens, and then the products are released. So in this particular example, we have a substrate that looks like this, and by the end, they're split up. And again, that's just an example. Now, what's important about enzymes is that they are reusable or recyclable. So as you can see, the enzyme at the end is still okay. It's not like it got used up. And so you can use it more than one time. So going back to our analogy of starting a fire, it would be more like using a lighter, because a match you can only use once. But in real life, enzymes can be used multiple times. So that would be similar to a lighter, where you can use it several times before it runs out. And so enzymes are extremely important for catalyzing these, reactions that are happening in living things. And not only that, enzymes can be regulated. We can turn them on and we can turn them off, which is really great because, for example, if you don't want a fire, you have the option to turn off the lighter. If you want the fire, then you can turn it on. And so just like that, enzymes can be turned on and off as well. So enzyme activation is turning an enzyme on. Not all enzymes, by the way, can be activated. Some enzymes are just always on, but many enzymes have to be turned on. And so we call this activation. And one way to activate an enzyme is to have a cofactor or coenzyme. So a smaller molecule that binds to the active site of the enzyme. And together, they're going to do the chemical reaction. Now, by the way, whenever a cofactor slash coenzyme is bound to the main enzyme, they together are called the holoenzyme. And so that's one way. The other way is through allosteric activators. And these are molecules that bind to your enzyme, but somewhere else on the active site. So here's the active site. The activator actually binds somewhere else. And then now the active site is available to do the chemical reaction. So the word allosteric means not at the active site of the enzyme. Or I guess, in other words, you can say allosteric means somewhere else. And so that's how we activate enzymes. And guess what? We can also turn off enzymes. So enzymes can be turned off using inhibitors. And there's two types. There's competitive inhibitors. They basically bind to the active site and block it. And so that's it, right? If you block the active site, then the enzyme can't function. Or there's allosteric, also known as non-competitive inhibitors. They bind to the allosteric site, which is, again, somewhere else on the enzyme. So here's our enzyme, here's its active site. The inhibitor is somewhere else, and that prevents the enzyme from doing its job. So those are two ways that we can inhibit. And inhibition is extremely important because, of course, you don't want an enzyme to be active all the time. Again, you don't want your lighter to be on all the time, right? And so inhibition is really important when we're talking about metabolic pathways. So this is an example of a metabolic pathway where we have one enzyme doing one job, and then the second enzyme does another job, and it's kind of like an assembly line. And so in this assembly line, once you get to the end product is actually the one that inhibits the first enzyme, which stops the entire pathway. And so this is a really efficient way for cells to regulate their metabolic pathways because they have the metabolic pathway, it's active, and then once they're done producing whatever it is that they need to produce, that product is what actually inhibits the cycle from continuing, which is super efficient. And so that's all I have to say about enzymes. And the reason why we're talking about enzymes is because, again, they play an important role in cell metabolism. And so in a few other videos, we're going to be talking about cellular metabolism, which is essentially how cells break down molecules and also build molecules. So when a cell breaks down a molecule, we call that catabolism. So if we have something like a sugar and we break it down for energy, that is catabolism. Or the opposite, let's say we are building molecules and storing energy, then that is called anabolism. So catabolism is breaking down, anabolism is building. And guess what? Enzymes are going to play a very important role in both of these processes. So this video was just kind of an intro to enzymes, and then we'll talk about metabolism in the next few videos. Hope this was helpful, and I will see you in those videos.